Okay, so we're going to pick up here on neuron classification. We said that neurons are classified in several different ways, like sensory neurons, motor neurons, and interneurons. Sensory neurons are the ones that can help transmit sensory information from your periphery receptors to your central nervous system or your brain and spinal cord. Motor neurons are the efferent division, and they carry information away from your brain and spinal cord out to things like smooth muscle, cardiac muscle, glands, and skeletal muscle. And that can actually help transmit information to other organs and tissues of your body. That's the motor neuron or efferent type of neuron. In between the two, you find interneurons. Interneurons are typically multipolar neurons, and they're highly interconnected, so they're really good at processing information. Now, what we see on this slide is a typical feedback loop with your spinal cord. So right here, we see sensory neuron, and it's going to pick up information about, like, let's say, stretch on your skin or something, right? And now that information can get sent along its sensory or afferent neuron towards your spinal cord. Hey, if the cell body's off to the side here, what type of neuron is that? Unipolar. You got it. And so that it keeps going, the axon keeps going, and then it synapses on an interneuron. This interneuron can help process that information, and then also synapse or connect to a motor neuron. And that motor neuron then can actually send information out along its axon to some skeletal muscle, which can maybe like, contract and then, scratch that itch or something, whatever you might have had on your shoulder. So this is actually just a typical feedback loop involving your spinal cord. So we've got a sensory neuron, interneuron, and a motor neuron. But you can actually multiply this on the scale of millions of neurons, and those would be more like complex neural networks, which you'd find in your spinal cord and brain. And those can be used for things like emotional processing, walking, talking, like even like higher order behaviors, which is actually kind of cool. Now, what this slide shows you guys is just a description and a summary of what we just talked about. So structurally, we got unipolar, bipolar, and multipolar neurons. Functionally, we got sensory, motor, and interneuron. So what type of neuron has lots of processes? Multipolar, right? What type of neuron has one axon, one dendrite? Bipolar. And then what type of neuron has one long, slender process? Unipolar. What's the most common neuron in your body? Multipolar, good. Which one's found in sensory systems? Bipolar. It's the bipolar. So special senses like your olfactory system and eye. Okay. And then unipolar, you find also in sensory neurons, but these are mostly going to be involved with reflexes, like detecting stimuli like chemicals and stretch and that kind of stuff. Now, functionally, we have sensory, motor, interneuron. If we say a neuron is afferent, what does that mean? Sensory, right? If it's sensory information's going to the brain and spinal cord. Good. If we say a neuron is efferent, what does that mean? It means it's motor, right? And information is being carried away from the central nervous system to a peripheral tissue, like a skeletal muscle, smooth muscle, cardiac muscle, gland of some sort. Good. And then what's the function of interneurons? In between sensory and motor neurons. Good. They're involved in information processing. Very good. And so what's the most common type of neuron again? Multipolar. And also we have a lot of interneurons. And so most interneurons are multipolar. And so these kind of go hand in hand. All right. Cool, guys. Good job. So what we're going to do next is move on and talk about the types of glial cells. Remember, glia outnumber neurons 10 to 1 in the brain and spinal cord. So they're the most abundant cell type in your central nervous system. We also call these neuroglia, and they're found in both the central and peripheral nervous systems, but not all the same cells are found in either, right? You have some cells that are specific to the central nervous system, others that are specific to the peripheral. And they're smaller than neurons, and they're capable of mitosis or division. Because they're capable of cell division, these are the ones that are more likely to become tumorous. So when you think of like a brain tumor, most often those brain tumors are made of glial cells, not neurons, because neurons don't divide that commonly or frequently. Glial cells do divide. So when you think about like brain cancer or brain tumors, it's most often a glial cell tumor of some sort. And there's different types of glia that can become tumorous. Now, glial cells are also more numerous than neurons, so they outnumber neurons 10 to 1. And their function is to protect and nourish those neurons. They're like the support team of a neuron. And this is kind of what they look like, like an artist rendition of the brain tissue here. So what we're trying to see here, guys, if you can like cut a piece of tissue out of the brain, zoom in on it, And then look at this sort of like jungle monkey mess of cells and tissue. Like when I look at this, it looks like a bunch of like cells like swinging on vines and stuff like that. And so these are neurons, right? And these are the dendrites of those neurons. And then we got this axon that continues out into space, right? But check this out. There's actually a cell that's actually wrapped around these axons. What? How is there a cell wrapped around these axons? Well, it's one of the cells that forms the myelin sheath. So it's actually involved with insulation, okay? It actually helps speed up action potentials, which is actually pretty cool. Now, we also have other cells nearby. Like, look at this one hugging a blood vessel. That's what I mean. Like, to me, it looks like a jungle or something. Like, this is like some sort of like sloth, like grabbing onto a tree branch or something like that, right? That's what my brain sees. I don't know why. But this is actually an astrocyte. And astrocytes surround blood vessels. They actually help form something called the blood-brain barrier, or BBB. We'll talk about this in a little bit. Now, what they do is actually help prevent molecules in the bloodstream from making it into your brain tissue. And that's one of the functions of these astrocytes. Now, we also find microglial cells in brain tissue. And microglial cells have an immune function. And they're involved in helping to clear out debris. In fact, microglia are a lot like macrophages. So what was the function of a macrophage? You got it. The big eater. Good. So microglia are like the macrophages of your brain tissue. And then these ones right here that are ciliated are called ependymal cells. Ependymal cells actually make something called cerebrospinal fluid, or CSF. It's a type of brain fluid that your brain floats within and it's nourished by. Okay. Let's talk about these, you guys. So we have astrocytes, ependymal cells, microglia, and oligodendrocytes. This is in the central nervous system. Okay. Now, astrocytes are large, very numerous, and they surround neurons as well as capillaries. They help form the blood-brain barrier, and they regulate tissue fluid composition. Astrocytes also provide support to your nervous system and also help guide the development of your brain. They replace damaged neurons and assist with development of your brain, and also help regulate synaptic transmission. So astrocytes are very important. In fact, there's a research study up in Fort Collins, a research group. They're studying the effect of astrocytes on consciousness, like your ability to be self-aware. And it appears that individuals that have issues with astrocytes also have difficulty with consciousness. So it's interesting that we believe that neurons are the only ones that are involved with communication. But there's some evidence to suggest that astrocytes also regulate communication, and they may also play a role in consciousness or self-awareness, which is actually pretty cool. Now, these ependymal cells you find lining the spaces inside your brain. We call them ventricles. And these have a lot of cilia, which actually are involved with making cerebrospinal fluid, which is basically like highly filtered blood plasma. And they also help move that cerebrospinal fluid around your brain to help form a little current that washes your brain in this fluid and helps nourish the brain tissue, but also helps to remove wastes from your brain tissue. And we'll talk about this in a little bit. How about the microglia? What was their function? You got it. Get rid of debris because they're like the macrophages of your brain tissue. Good. And then oligodendrocyte. Oligo means many. Dendro means kind of like extension, right? So oligodendrocytes have many branches, and oligodendrocytes are the ones that make myelin in the central nervous system. You got it. I know it's weird. Like people like flip a U-turn right here. Like, what are you doing? Oligodendrocytes make myelin in the central nervous system. So the myelin sheath was what wraps axons, and it actually acts as a really good insulator, which helps increase action potential speed. Now, the peripheral nervous system has satellite cells and neurolemmocytes, which we'll talk about in a little bit. So the astrocytes are the ones that look like stars, right? Hence the name astrocyte. And they're the most abundant glial in the central nervous system. Now, they make the blood-brain barrier, which is a protective barrier that prevents substances in your blood from getting into your brain tissue, because there are things in blood that could damage your brain tissue. For one, bilirubin, which is a breakdown product of hemoglobin. It looks kind of a yellowish-green color, and when it aggregates in your skin or sclera, it makes them look more yellow, which you guys know as jaundice. We learned about it back in skin. That molecule is toxic to brain tissue. If it crosses into your brain from your bloodstream, it will kill your brain tissue. So in order to prevent that from happening, then we don't want bilirubin to cross, right? So luckily we have a blood-brain barrier that prevents things like bilirubin from getting into your brain tissue and potentially damaging the tissue. Astrocytes also help regulate tissue fluid composition, like the composition of the fluid you find in your brain. It helps regulate like the concentration of things like sodium and glucose and potassium in our brain tissue. It also helps regulate synaptic transmission, so it helps neurons communicate with each other. We talk about how astrocytes may be involved with consciousness. They help form a structural network. They help replace damaged neurons. And they also aid in the development of neurons because astrocytes actually guide the way during neuronal development. As your brain's growing in size, astrocytes actually have a long process. They stick out, it almost looks like a fireman pole, right? Like little poles that like probably they use in firehouses. Do they use this still? I don't know.
Yeah,
They do?
yeah, they're all like, I mean, yeah.
That's cool. That's really cool. I want one of those.
All there.
Like, could you imagine? I live in an apartment. Like, it's not really cool. No, it would be cool to have one though.
Like a stripper pole.
Yeah, exactly. No, it's a professional dancing pole. So, you know, these astrocytes have something that looks like a professional dancing pole that extends out into space, and then and then neurons walk along that pole while your brain's growing. How weird is that? So astrocytes actually kind of help guide the way by sending out these little poles into space, and then neurons walk along those poles as your brain's growing. So actually these astrocytes guide the way, which is pretty cool. That's how they assist in neuronal development. Now, what we see here is the astrocyte in green. And it's something that's surrounding this capillary and connecting to the neuron. So it helps form the blood-brain barrier because it surrounds your blood vessels. It also helps provide nourishment and regulate the tissue composition of your brain tissue by being close to neurons. It also aids with communication because you find this at synapses, or basically where neurons connect. Now, ependymal cells are kind of like a ciliated cuboidal epithelium type of cell that line the ventricles of your brain. This sounds weird, you guys, but there are spaces in your brain, like large fluid-filled spaces in your brain tissue. You've probably never seen these before, but if you cut a brain in cross-section, you'll notice there's big spaces in the tissue. Like it's not like your brain's solid. There are large fluid-filled chambers inside your brain. That fluid-filled chamber is called a ventricle. It's basically the space by which your brain develops around. We'll get to this a little bit later. Now, what lines the space inside your brain is not a true epithelium, but it's like an epithelium, and we call this the ependymal cells. Ependymal cells are like a ciliated cuboidal epithelium. What was the function of cilia? Yeah, to move stuff, right? Kind of wave around. They can help move material across the cell surface. And then cuboidal epithelium is really good at secreting. So you find it in glands. But here it's secreting cerebrospinal fluid into the brain space, the ventricle, and then the cilia of these cells wave it around. That way you get a kind of a nice current of fluid movement inside your brain, which does two things. It helps keep your brain buoyant because your brain is several pounds and it could actually crush under its own weight. But luckily within our skull, our brain floats in a fluid layer called cerebrospinal fluid. And it also helps nourish our brain because there's nutrients in cerebrospinal fluid. So the glial cells here, the ependymal cells, actually make CSF, and you find these in the spaces of your brain. So here's that space. You know, it's zoomed in, so you can't really see much of it. But what lines the space inside your brain are these ependymal cells. They have lots of cilia here that can wave it around. They can move that fluid around. And these ependymal cells take fluid from your brain tissue and then basically convert that into cerebrospinal fluid, which in composition is a lot like Gatorade. That's what I like to say. So CSF is a lot like Gatorade, cerebrospinal fluid. So what's Gatorade mostly? It's actually mostly water, actually. It's mostly water, right? Then it's followed by sugar, then electrolytes. Electrolytes would be things like sodium and potassium and magnesium and calcium. That's basically what cerebrospinal fluid is. It's water, sugar, electrolytes, Gatorade, but it's for your brain. It helps your brain float, and it helps nourish your brain tissue, and keeps it clean, I suppose. Now, microglial cells were like the macrophages of your central nervous system, and these ones are involved with helping to remove any kind of debris that might have accumulated in your brain tissue. And so you think about like brain death or parts of your brain kind of dying off, or if you have debris in your brain, these microglia can actually help remove that. And they clean it up by engulfing it, digesting it, and then taking those little bits and dumping it back in the tissue where it can float back in your bloodstream, and it can be recycled by your body or eliminated through like feces or urine. Now, these microglial cells, as the name implies, they're small, when they're inactive. But when they activate, they get much larger, and they have the ability to engulf things like microorganisms or debris inside your brain tissue. And so this is like the immune cell for your brain. Now, oligodendrocytes you also find in the central nervous system. These things actually can wrap themselves around axons, and they do this multiple layers around an axon. They form the insulation, which is the myelin sheath. Very good. So oligodendrocytes produce myelin, which is an insulator for electrical activity in the axons. So if you look here, this is an oligodendrocyte, and it's wrapping around the axons here. Now, with multiple wrappings, it forms a nice thick layer of tissue or cell that basically helps function as an insulator, and it helps current from leaking out of the axon. That way, electrical currents can actually spread more quickly down the axon with a myelin sheath. In the absence of myelin sheath, or if these oligodendrocytes didn't make as much of it, then you have difficulty transmitting electrical impulses around your body.
It's like if you were to have like a electrical cord that you plug into the wall and it didn't have the plastic tied around it, it could go everywhere.
Yeah, exactly. You got it. Yeah, if you had like exposed wire, then you could have electricity kind of spreading in weird ways. Yeah.
Is there some reason why parts of the axons are exposed between those little segments?
Dang, you got excellent questions. I love this. Okay, you raised a great point. Why are the little gaps? You'd think that like if to have a good insulator, you'd want to have insulation along the whole dang length of that axon. So why put a gap there? Well, we'll come back to this when we talk about action potentials. But the reason why you need a gap is that you need to regenerate that electrical stimulus at those gaps. They're called nodes of Ranvier, and they're regenerated at each of these little nodes. Okay. Now, what I mean by regenerated is that they're replenished. Like, so what happens is that electrical currents can spread from this node really quickly through the sheath to the next node. Then it's regenerated, and it spreads quickly to the next node. And it's regenerated, and it spreads quickly to the next node. And this way you don't lose a signal. Otherwise, if you only had insulation along the whole length, your current would leak out, but you couldn't actually regenerate the action potential, which means your signal could get lost over time and distance. But we'll come back to that. I know that's not the best explanation, but I mean it'll make more sense in a little bit. So what about the glial cells of your peripheral nervous system? Well, the peripheral nervous system is everything else other than your brain and spinal cord. So this is like peripheral nerves and ganglia. And there's two main types of glial cells here. We got satellite cells and neurolemmocytes. The satellite cells you find surrounding neurons in the peripheral nervous system, and they're like the astrocytes of your PNS. And the neurolemmocytes form the myelin sheath around axons in the peripheral nervous system, or PNS. Now, this is the cell body here in the peripheral nervous system, you guys. These are satellite cells that surround the axon. They surround the cell body. They basically just nourish the neuron here. And then over here we have an axon, and these have what are called neurolemmocytes. Each one of these little blue things is one neurolemmocyte, which is different from the central nervous system because we had an oligodendrocyte could myelinate many axons at the same time, whereas a neurolemmocyte can only myelinate one axon at a time. And even then, it can't do a complete axon. It can only do like portions of the axon. So these are neurolemmocytes. You have a question?
Yeah, what did you say that satellite cells?
Satellite cells are like astrocytes, so they kind of help nourish the neuron. And then the neurolemmocytes form myelin in the peripheral nervous system. What if someone had a disease of oligodendrocytes? Would they lack myelin in the CNS or PNS? CNS, right? Or neurolemmocytes? Peripheral nervous system, exactly. So that can occur. So there's different types of demyelinating disorders where you can lose myelin in specific parts of your nervous system, like only the brain and spinal cord or only the peripheral nerves. It just depends on the disease. And we'll come back to these kinds of things in future classes. Now, the neurolemmocytes are Schwann cells, and these are actually going to be the things that form myelin in the peripheral nervous system. They wrap themselves around axons to form, like taper on electrical wire. They produce myelin, which is an insulator, just like oligodendrocytes do. And they have the same structure and function as oligodendrocytes. So what they look like then is each one of these is a neurolemmocyte. It's wrapping around the axon, forming a nice little myelin sheath, which conducts action potentials more quickly. That's the function of myelin there. So the whole idea of myelination then, like the process of forming myelination, is to affect the ability of nerve cells to transmit their action potential. So if an axon has more myelin, then it can transmit action potentials faster. If an axon has less myelin, it will transmit action potentials more slowly. Okay, so myelination is the process of forming that myelin sheath, and it's formed by neurolemmocytes in the peripheral nervous system or oligodendrocytes in the central nervous system, and it serves as an insulator so that currents don't just leak out of the axon. So what this process here shows you guys is what myelination will look like. So here's a cell. You can tell because there's a nucleus and there's cytoplasm, and here's the membrane. And what the cell does is it basically just starts to wrap itself around the axon many times, over and over and over and over and over. And by wrapping itself a lot of times, it forms this nice insulation or protective layer, which is the myelin sheath, and it prevents currents from spreading out